So it's a great pleasure to be back here in Tübingen. I have very fond memories of Tübingen. I not only completed my scientific education here, but also my family. And I'm also very pleased that so many of my co-authors can be here today who really not only contributed to, who helped create the work that I'm going to present to you today. Now, color is the process that starts with the activation of the cone photoreceptors in the retina. And these excitation patterns then get conveyed to the visual cortex, where they're interpreted as color sensations. That's not really what I'm going to talk about today. What I want to talk about today is rather what color information is used for. And in my view, this is really to recognize objects quickly and to remember them better. Now, before I get into that, I'll just quickly review and say a little bit about the cones. So, colorimetry has really been the major success story in the field of color vision in the last 100 years. And the introduction of the CIE standard observers has really helped to make life easier, not just for scientists, but also for business and industry. And it's not by chance, one of the very first books I read about color vision was by Dean Chudd, Color in Business, Science and Industry, and science is only in second place there for a good reason. And what I've illustrated here is I've taken a Macbeth color checker into the botanical garden here in Tübingen, and then we took a measurement with a spectral radiometer of the red patch here, and the light that gets reflected from that patch can be characterized by its spectral wavelength distribution. And you need basically several hundreds of numbers to characterize that distribution, and it's very difficult to deal with this distribution because usually it's hard to tell what color this corresponds to in real life. And what helped enormously is the knowledge that we obtained about the cone spectral sensitivity functions, and basically that got investigated starting with Maxwell and Helmholtz. And there the big achievement was to find out that basically 3 numbers suffice to characterize the effect of the light that's characterized by these many hundreds of numbers on our visual system. And that made life easier for everybody. And it's relatively straightforward to, when you have these 3 numbers, the cone excitations, to say which one of these patches it corresponds to, depending on how much sort of the blue, green and red cones get excited. Now, at the scientific level, there's been at least as much, if not more, success. We now know these functions really up to 3 decimal places. And you can see here the cone absorption spectra for the short wavelengths, middle wavelengths and long wavelength sensitive cones, usually just called blue, green and red cones. And what you can see here is that they actually cover the whole wavelength range. They're not sensitive to little parts of the wavelength spectrum corresponding to red, green and blue. And in particular, the spectra of the M and the L cones are highly overlapping. And one of the reasons for this large overlap is that they only recently differentiated from a common ancestor. And so about 35 million years ago, our monkey ancestors in the African jungle were dichromats, and they only had one of these cones, and by some random mutation, the 3rd cone type evolved, and that made it presumably easier for them to detect ripe red fruit amongst green leaves, and sort of eating the fruit was easy. The fruit is easier to digest, and that gave them spare metabolic energy. Now, this evolutionary process and the genetics of these of the 3 different contacts is extremely well investigated, and Jay Knights and his colleagues recently even managed to insert a third cone type into dichromatic monkeys by gene therapy. So it's really just amazing progress that has been made in this field. I just want to illustrate you quickly with this picture here that was not taken in the jungle in Africa, but in my backyard. And what you see here, and what you can easily see here even in this black and white image, is sort of a raspberry amongst some leaves. And when I show you a version of this image that simulates people who either lack the L cones or the M cones, they can still reasonably well, in this relatively straightforward case, detect the raspberry. But you will agree that it's much better visible here in this full color image. And I think what I'd like to point out, and what's particular about this image, is that only here you have a chance to detect the second raspberry that's slightly occluded. And that image also demonstrates to some degree one of the main messages I'd like to convey to you, that color vision really helps us to recognize and detect objects more easily and effortlessly than it would be possible if we don't have at least the third color dimension. So, to sum up what we know about cones, we know a lot about cones. We can measure isolated patches of colors very precisely, and we have an excellent idea why primate color vision evolved the way it did. What more could anybody ask for? Well, that's the question that Otto Grüsser and Theo Landis asked themselves in 1991, and they came up with a lot of more questions. So this diagram illustrates the processing of color information in the human brain. And you can see what I've showed you now is all been in the retina, the processing in the cone photoreceptors, and the second stage of processing, the color opponency, is also quite well known, and it also still happens in the retina, in the retinal ganglion cells. But from there on, much less is known about these different processing steps. And I've colored in this diagram that was originally black and white, and basically the red parts here is what we know really well, and the blue parts are where we don't know much about. And these parts here, surface color, contours, or color and movement, those have been studied a lot in the 80s and 90s, and we gained some knowledge, but other aspects are still way in the dark blue. And interestingly, during the last decade, there's been a lot of attempts and progress has been made to relate color categories and color names and relation to language and memory to this earlier mid-level representation of color in the brain. Some of these other aspects of color that are not so well understood yet are illustrated in this slide. Now, if you look at this collection of black and white images, then after looking at each one of them, after a while you figure out what's shown in each one of the images. But when I show you the same images in color, you'll have no problems to say much quicker whether there's sort of pieces of fruit or greenery or landscapes or animals or just about anything else in these images. So this really shows that color helps you recognize things more quicker and effortlessly than would be otherwise possible. And what's also relevant here is that for some of these images the colors might be aesthetically pleasing to you, or they might lead to an emotional response. These are aspects I'm not going to talk about, but there are other speakers during the course of today and tomorrow who will deal with these aspects where we really don't know very much yet how it's really being processed. Now, what I would like to look at when in this diagram is really how color helps us to recognize objects and how color helps us to remember objects better. And this is again illustrated in this photograph here of some flowers. And I've decomposed this image into a chromatic part that is isoluminant. Basically, I've kept the color at each pixel and set the luminance to the exactly same average value. And here is the same image in black and white. And what you can see immediately is that it's much easier to segment the original image or even the isoluminant colored image. It's easy to tell the different petals apart, to tell the petals from the leaves and from the background. Whereas this is all very difficult or even impossible in the black and white image. But I'm not only going to show you demonstrations here. Basically, what I just said is that color helps us to recognize objects more quickly and to remember them better, is the result of some experiments that I'm going to show you now. And these were actually done here in Tübingen together with Felix Wichmann and Ted Sharp. And we did the most simplest experiment that you could possibly think of. So we just took a set of about 50 images and we presented them to our observer one by one. Every 5 seconds, one image, and they were shown for various durations. And then, after these 50 images were presented, there was a break for a couple of minutes, and then we presented the same 50 images interleaved with 50 new images that the observers hadn't seen before. And for each image, they just had to say, I've seen this before, or I've never seen this before. So it's an extremely simple task. And half of the images were shown in color, and the other half was shown in black and white. And the images were basically from 4 different classes of images. So the first class of images was green landscapes from around Tübingen, and they basically, the color was very homogeneously green throughout the image and also didn't differ very much between the images. The second category was these flower images, which had sort of many different colors in them, with flowers and leaves. Third category was these rock landscapes from the southwest of the USA, and they were mainly reddish brownish, but had many different shapes. And then the fourth category was just snapshots of including man-made objects. Now, the results of this experiment are shown here. So the exposure duration with which the images were presented is shown here on the x-axis. So it varied between 50 milliseconds, a very short flash, up to 1 second. And the proportion of correctly identified images is shown on the y-axis. And one thing you can see immediately is the longer the image gets presented, the better people are in remembering it. That's very trivial, but it's reassuring. The second aspect, which is important here, is that the curve for the color images is above the curve for the black and white images. So people do remember the color images better than the black and white images. And one thing that was stunning to us from these experiments was that was the case even for the very short presentation duration of 50 milliseconds. And I'm going to get back to that point in just a minute. But I'll first like to show you data for the same experiment from a group of color deficient observers. So those were dichromats who are lacking the red-green color opponent system, and it's very common in the male human population at around 4%. And Ted Sharp at that point in Tübingen had this large sample of dichromats which he used to get very precise measurements of the cone spectral sensitivities. And when we did the experiment, we basically couldn't believe our eyes. What happened was that the performance, the memory performance for the color deficient observers was exactly the same as for the trichromatic observers, and they also showed this advantage of color, which there's of course, what you have to keep in mind is that these people are not color blind. They only lack one dimension of color. They still have color information available. And this slide here is from the seminal work by Hans Brettel, François Vigneault, and John Mullen, and it illustrates what the world might look like for these color deficient observers. So here's again, there's very popular images of flowers, and then what it would look like to a protanop, deuteranop, or a person basically lacking the S cones. This is very rare. But our observers that we used in these experiments would see the world like this or like that, and you can see that there is still color information available that can help you segment the scene and to tell basically the flowers from the leaves and the background. And observers, the dichromatic observers, seem to really be able to use that information. Interestingly, there's been some experiments in Andrew Darrington's lab in England where he presented trichromatic observers with images like this, and there he found that there was no color advantage for memory when doing that. That is, what this means is that these dichromatic observers really learn over long periods of time to use the residual color information they have available. And it's not directly and immediately available to trichromatic observers. Now, I'd like to get back to the aspect that I mentioned before, namely the color information is processed very quickly. And of course, we wanted to know what's going on in these first 50 milliseconds. So, of course, we could have presented the images even shorter, but our observers were already, with this very short presentation duration, they were close to guessing, and it would really have frustrated them. So we used a different paradigm to study this initial period. We used a so-called match to sample task, and it's again very simple. Here's an illustration of what happens on each trial. We present an image, a target image, and it's followed by a mask. And the mask has the effect basically of wiping out the residual of that image in visual cortex, so that the target image really only is available for the short period it is presented here, between 8 and 64 milliseconds. And then after the mask, we show two images, the target image together with a distractor. And the observer simply has to say whether she or he has first, which one of the two is the target image. And the mask was chosen really to be very similar in spatial and chromatic aspects to distractor and target, so it doesn't have any diagnostic information. And in this experiment we chose again the conditions where the images will be presented in color and then tested in color, or presented in black and white and tested in black and white, but also we added a new condition where the image would originally be presented in color, but then the testing would be done in black and white. Now, why does this new condition help us to gain further information about the processing of color for recognition and memory? Well, you can see that any difference between these two conditions couldn't be due to the initial processing of the test image. It must have to do with the testing phase. That is, it could not be due to sensory factors, but it must be due to cognitive factors. For example, if the observer sees this image and recognizes that there's something purple in there, she or he can later use that information when viewing the test, because there is also something purple in here. But when the testing is done in black and white, that doesn't really help anymore because there are no colors here. So any difference between these two conditions is due to the representation of the information in memory or due to cognitive factors. Whereas any difference between these two conditions must be due to sensory factors, due to the initial presentation and processing of the image. So if color helps us here to segment the different parts of the images quicker, then we can use this quicker processing to recognize what's on the images, and we can use that later on, even though the testing is done in black and white. But that additional sensory processing won't be available here when the presentation is done in black and white. So differences between these two conditions are due to sensory factors. And now we can look at the data and see whether the advantage of color that we hope to replicate from before is due to sensory factors or due to cognitive factors. And the results are shown here. And interestingly, at the short presentation durations, so here again presentation duration is on the x-axis and the proportion of correct identifications on the y-axis, and you can see when the images are presented in black and white and the testing is done in black and white, people are at about 65% correct, and they're about 7 to 8% better when the presentation is done in color. And it doesn't matter whether the testing is done in color or in black and white. So the difference here must be due to sensory factors, and that color helps us to better recognize the objects and to segment the images. But when the exposure duration increases, and here at 64 milliseconds, we still find a difference between black and white and color, but now the difference is only there if the images are presented in color and tested in color. So this difference here is due to the memory representation. And the interesting thing is that for intermediate presentation durations, we get a gradual transition. So initially, the images presented in color are processed better, but then eventually sort of the grayscale processing catches up. It just needs a little bit longer. But then, of course, when the testing is done in black and white, it cannot reach the same level of performance. So what these data really show is that color helps us to recognize objects quicker and to remember them better. And the question that remains, of course, is how can color be so fast? Because the prevailing theories of color processing, and I think most of them can be characterized as a sort of a coloring book theory of visual perception, where when we have some visual input or visual image, first a luminance-based system comes and draws in all the contours, and then after a long time the color system comes in and fills in the surfaces. And if that's the case, then we would expect color to make the contribution of color to be rather slow. But that's not the case, and it seems to be relatively fast. And in fact, recent work from neuroimaging has shown that there's actually a very brisk response to color in primary visual cortex, where there are mostly edge detectors for extracting contours. And some work doing electrophysiology from single units in primary visual cortex from Bob Shapley's lab has shown that many neurons there are actually sensitive to edges that are defined either by luminance or by color. So we wanted to see whether that information about edges and color is really out there in the world. So to study that, Thorsten Hansen and I looked at a large collection of calibrated natural color images that come from McGill University, from Fred Kingdom's lab. And what we did was we just analyzed these images using a standard edge detector. And here's an example image, and we decomposed that image into a luminance image, and here into a red-green image, the component that's extracted by the red-green retinal ganglion cells. And then we ran just a standard Sobel edge detector on these images, and you can see the output here, the luminance edges, and here the red-green edges. So first of all, there are red-green edges here, and then we created a histogram. For each edge in the image, we indicated the strength of that edge in the red-green direction and the strength of that edge in the luminance direction. And this is this histogram, this joint edge histogram for the image that is shown up here. And some of the edges are marked by numbers here and there. And you can see, for example, here, edge number 6 is an edge that has a luminance contrast and a red-green chromatic contrast. It's the border between the petal here and the background. But there are edges also that are purely isoluminant, that have no luminance contrast. Edge 1 and edge 2 are examples of that. For example, here, this border between the petal and the leaf happens to be isoluminant. That is, there's no luminance contrast involved. And because most of the edges here have some luminance contrast and some chromatic contrast, of course, just by chance there happen to be some of these edges where by accident the luminance contrast in the two objects matches perfectly. And those are cases here. There are also some edges that are pure luminance edges, edge 3 and edge 4. And you can see edge 3, for example, here, that's within a leaf. And it's clearly obvious that this is just a shadow edge. That is an edge simply due to lighting. It has nothing to do with the surfaces or objects. And clearly this, if the light source moves a little bit or so, then this edge will also move. It's not an edge that indicates to us an inherent property of the object. So it's more of an accidental edge. And so these kind of edges that are purely caused by luminance and where the color remains the same, they're basically mainly due to the physics of these of shadows and lighting changes. And they might not be as useful in extracting object information. But this histogram here is actually quite typical for the whole collection of objects. So here you see this joint histogram over all images for the red-green and luminance and for blue-yellow and luminance. And you can see that nearly all of the edges sort of have luminance and chromatic information combined. And there are very few that are purely isoluminant or purely on the black and white or grayscale axis. So what this indicates is if we want to have reliable detectors of edges in the real world in our visual cortex, then these detectors should be sensitive both to luminance and chromatic changes at the same time. And that seems to be the case in the primate brain. So we're very well tuned to these detectors. Now, in this image you've already seen that maybe the edges that were indicated by color here correspond a little bit better to what we would call meaningful edges in this scene here. So Thorsten Hansen and I went a step further and we compared the edges that our color luminance edge detectors would find to human marked edges. And there, in computer science there's a large database where up to 40 observers have labeled the edges in images like this one. And we compared these human marked edges to the edges that were extracted by a luminance edge detector or by a color edge detector. And then we compared sort of luminance only to color only and to color and luminance. And the interesting thing that we found was that actually if you have luminance edges and you add color information, that leads to an improvement in more than 95% of all the images. That is, people get better if you add color. If you take the color information and you add luminance, there's basically no improvement. So, which might just be due to the fact that the luminance edges might just, in many cases, signal shadows or other accidental lighting features. So color information is really extremely useful in this task of detecting edges. And lots of psychophysical experiments have shown that human observers are also very good in judging the orientation of chromatic edges. Now, the color information about edges here, that is actually part of the processing that is so successful in making, in helping us to recognize objects more quickly. Now, when we get to the point about memory, remembering things better, there's an additional complication. And the additional complication is that just remembering the cone excitations won't do us much good. So here's again the diagram where we have the isolated patches and the cone excitations that go along with it. But the problem is that the same cone excitations can lead to different color appearances, and different cone excitations can lead to the same color appearance. Now, here you see the 4 patches, blue, yellow, green, and red. And here at the center, you see 4 patches that probably look relatively similar, blue, yellow, green, and red. But what's happened here is that I've simulated an illumination on these patches, and in fact all of these 4 patches have the same neutral distribution of light, and therefore lead to the same neutral level of cone excitations. So the way the color percept arises here is that basically here a surface that appears blue under neutral lighting is simulated under yellow light, which basically makes the spectral distribution neutral, but we discount this illumination. Our visual system is color constant. We perceive the invariant properties of the object, of the surface reflectance, and that's why this appears blue to us and this appears yellow to us. I'll indicate this here. It will maybe become a little bit clearer in this slide. Here I've taken a set of objects, sweaters, candles, this booklet, just random objects. And I've illuminated these objects with 4 different illuminations. The illuminations were created by putting colored filters in front of the windows. So it was a full field illumination. And then I took pictures of these objects under each of the illuminant with a constant white balance setting of the camera. So basically what you see here might be similar to the cone excitation patterns that arise from the objects in that scene. And now if you basically cut out little patches from the objects under these 4 different illuminations, you can see that the color of these isolated patches here actually varies quite considerably. There's this sweater that appears sort of light greenish, actually changes color here from up to gray brown or to blue. So it's quite a massive change. And what this shows is that if we would not have color constancy, if our visual system would only measure cone excitations, then basically objects would change their color all the time, like shown the way it's shown here. And this is, of course, this would be a massive problem, or basically what it means is that we couldn't really go ahead and assign color names to objects. It wouldn't make sense to call a sweater green if it would be blue under a different illumination. And that is what I would call the paradox of color constancy, because in the real world, color constancy really seems to be given. Of course, we call a sweater blue, and it looks blue under all illuminations, except for some exceptions like monochromatic light or similar. So, but in the laboratory, there have been lots of measurements of color constancy, and what we typically found in the laboratory was very poor levels of color constancy, of like 20% or just a little bit better than no control. And that is really a contradiction because, well, as I said, in the real world we seem to be color constant, and in the laboratory we're not. And one paradigm, it was actually sort of one of the first paradigms that has been used to study color constancy, is this asymmetric matching technique, where basically one illuminant illuminates one side of a screen and another illuminant illuminates the other side, and most of the time these illuminants were simulated on a small computer screen, while people experienced some completely different illumination in the room. And using these relatively simple setups, people have found that there was just a little bit of a correction for color constancy, and Brian Wondell very nicely summarized this in his book Foundations of Vision, that when the illumination changes, color appearance changes less than it might if color was defined by the cone absorptions. So that there is a little bit of color constancy, but it changes more than it would if the nervous system used the best possible computational algorithms. So this means that it's not perfect. In the following years after these initial studies by Arendt and Reeves and many others, different paradigms have been introduced that actually showed that maybe color constancy is a little bit better than just 20%. And one of these paradigms was, for example, achromatic matching. And the nice thing about achromatic matching is that now we don't have two different illuminants in the scene. We just tell the observer to adjust a central the color of a central patch so that it appears gray, neutral gray without any color. And when observers do that, here is sort of a simulated change in illuminant towards bluish, and then you can again measure what observers adjust. But in these achromatic matching experiments, typically higher levels of constancy are found, and that probably has to do with the fact that there's only one illuminant in the scene, and the observers don't have to deal with two illuminants. So here we sort of get up to 50%, and David Brainerd, in a very nice experiment, has shown that if you put observers in a room with a real illuminant, that in the best case, with this achromatic matching task, they can get up to 80%. And furthermore, he has shown that if you take systematically the cues away that we know that contribute to color constancy, like the local surround, the average color in the scene, or the color of the brightest patch in the scene, then color constancy sort of gradually decreases. So all of these cues, in a sense, contribute to color constancy, and it's more similar to size constancy. We normally in the real world we're perfectly size constant, but if you go to a laboratory and close one eye so that its distance judgments are more difficult, then our size constancy decreases a little bit. And it's the same thing here with color. But what's still interesting, that this goes up to 80% and not to 100%, the way it does, for example, for size constancy. And another drawback, of course, with achromatic matching is that it's kind of a bit awkward to talk about color constancy when the only color the observers ever adjust is gray, and basically they make the patch to look as if it doesn't have any color. So we cannot do the same experiments with red or green or whatever. And at this point, together with Maria Olkon and Christoph Witzel and Thorsten Hansen in my lab, I actually thought maybe the important aspect about color constancy is that we end up with the same color name. That is, if we have a sweater and the illumination changes and I call this sweater usually looks blue, then it's good enough if I still call it blue under another illumination, and it's okay if the blue it's a slightly different blue. So that we call categorical color constancy. And the way we measured this was again with a single illuminant where we put colored filters across the window, and then we gave observers several hundred of these Mansell chips, and they just asked them to sort them into different bins, and they could choose how many different bins they would want to use. So usually observers would sort of put the red ones into a bin and the blue ones into a bin, and then we could actually measure sort of for each observer the categories and the boundaries between the different categories they use by looking at these bins. And we had them do the task under different illuminants by putting differently colored filters there. and then we express the degree of color constancy by checking how often they put the same two bins into the same two chips into the same bin. And the results are illustrated here, and you can see that on average the observers put the chips from the red category of Munsell chips in about 70% of the cases into the same bin. So that's the degree of color constancy here. And you can also see that it varies a bit with the hue, which is indicated here on the x-axis. For the greenish hues, our observers were basically 100% color constancy. They never put the greenish chip into a different bin. And one reason for that, it might actually be because the greenish category tends to be relatively white. And this thin line down here basically indicates a kind of baseline. That is, we simulated whether the chip would actually, under a different illuminant, shift into a different category, or whether people would put it into the same category even if they didn't have any color constancy. And this shows that basically for this greenish category, this problem of a different categorical, a different color category doesn't even arise very much. But in the end, on the average, we're sort of at around 80–85% color constancy. And after some more thinking, I thought, well, it's not really the case that we only keep the category information. We really do know what kind of hue a particular object has. We're better than just the category. And overall, if you look at many studies over many years, you can see the trend that I already showed you in the last few slides here, that initially in '86, in these early studies, color constancy was very poor, and then using different paradigms like achromatic matching, categorical color constancy, the measurement of color constancy seemed to have increased as our methods got better and more fine. And what this regression line that is put here on data that were compiled by David Forster in a very nice review in 2011 seems to indicate that it's basically color constancy is on the rise, and the prediction is that now there should really be a new method and a new experiment that really shows that we are 100% color constant. And that's what I'm going to show you now. So, and basically the experiment that we did was set up in a way so that observers would have all possible cues available to achieve color constancy. So and we used a memory-based paradigm to measure constancy. So observers brought in different objects. Each observer brought in one object that they were highly familiar with. And these are most of the objects. Some of the objects sort of had to be returned to the observers by the time we took the photograph. One observer brought a piece of the kitchen cabinet that she looked at very often, but she really needed it back home. And you can see that these objects cover color space really well. So we have all different hues. It's good coverage of the CIE, LAB color space here. And the task of the observer was to select a Munsell chip out of these 1,300 Munsell chips that they thought would best match their object. So if I brought in the orange sweater, I would have to pick the one chip out of these many that would best match my orange sweater. And basically the sampling of the chips is very dense, so for each object there are really a few chips in the neighborhood that are good candidates. But the crucial part about this experiment is that the object was never present when the observers made their matches, when they selected the chips. That is, observers brought in these objects and we shut them away in a closet for a few weeks, and the observers came to the lab. They came into a room which had these colored filters. This again is taken with a set white balance. If you come into this room with the colored filters, with the really pink colored filters, you don't even notice it. You notice it when you exit the room, but when you walk in, it looks like a regular room. And I've already shown you this slide indicating the changes that would go on in the color coordinates of these objects. And of course, if an observer is indeed color constant, then we would expect that she or he always picks the same chip to match the object, independently of whether the room is illuminated by a reddish light or by a bluish light or any of the other two illuminations that we had used. And we can then specify the degree of constancy by sort of comparing it to a match based simply on cone excitations. So if observers would use the cone excitations only, they would of course choose a very different chip indicated here, and then if they choose the same chip, this is 100% constancy, and if they choose a chip that's slightly off, we just project it on this line and express the degree of constancy by the relative lengths of that projection. And what we would expect here is that what the illuminant shift does is it shifts the color of the object in color space. In this case, for this greenish illuminant, the shift is of course in the greenish direction here. And for each object, there are two lines extending from the original location in the color space. One line is the shift of that particular chip, and the other line ends in a cross. That's the actual setting the observers made. And you can see that for most of these objects, except for this one here, the setting of the observer and the shift of the chip actually agree. And in many cases, in about 25% of the cases, they agree 100%. Observers do select the same chip. If they do select another chip, the distance between these 2 points can be expressed in CIE-Lab relatively well as the Euclidean distance, delta E, and here's a histogram of these delta E distances, and you can see that there's many cases where the same chip is selected, and on average this delta E distance is about 5 units, which is relatively near to discrimination threshold. So observers are really very, very good at selecting these chips. So for real-world color constancy is of course impossible to achieve for arbitrary stimuli and arbitrary illuminants. Just monochromatic light is just one example. But that doesn't seem relevant for everyday life. If you have natural objects and reflectances, a full-field illumination, and an observer who is really acting in the scene, then constancy is perfect, and we do know the mechanisms that can contribute to this task. And of course, color constancy is the prerequisite for memory. And I think in the last 2 minutes, I'm going to show you some even newer results where we were looking for the source of color constancy in the human brain. That is, we used functional magnetic resonance imaging to see where we would be able to extract color constant resources in visual cortex. And the way we did this was using a multivariate pattern classification approach. That is, in one condition, we presented our observers with chromatic patches that were presented in a neutral context. And these patches could either be yellow or blue. And in another condition, we presented our observers with patches that would appear yellow, but were colorimetrically neutral. And this is achieved, of course, by changing the illumination. So here, when the illumination is bluish, the central patches would appear yellowish, just because that's what yellow object would appear under this bluish illumination. And we presented these patterns interleaved. So here's a neutral context, blue context, neutral context, yellow context, and measured the brain activity in visual cortex while observers were looking at these patterns. And of course, the first thing we did was we trained a multivariate pattern classifier. That is, we looked at the brain activity in each voxel, and then we tried to predict whether the observer actually saw this yellowish patch or the bluish patch. And that has, of course, been done before by several other groups of people. And what is typically found is that if you train the classifier with one set of trials and then use different trials to test the classifier, that you can identify in these new trials in about 60 to 70% of all cases whether the observer has looked at a yellow or at the bluish patch. The crucial point is that now we want to test whether the classifier just tells us something about the color coordinates, the colorimetric condition, or whether the classifier really tells us what colors are perceived by the observer. And so in this case, we train the classifier again with the neutral context, but then we do the testing on those trials where observers had seen sort of the yellowish looking patch that has actually a neutral spectral distribution, or the apparently bluish patch that also has the same neutral spectral distribution. And when we do that, we find that in areas V3 and V4 in extrastriate visual cortex, the classifier would differentiate between the apparent colors just as well as for the colorimetric colors. So you can argue that in these areas color constancy is achieved, whereas that's not the case in V2, and it's only partially the case in primary visual cortex V1. And that, I think, actually agrees quite well with what we know about the mechanisms of color constancy that depend partly on local edge information that would be available in V1, but partly in averages over relatively large areas of the visual field, such as receptive fields in V4 might see. So to conclude, overall, I hope that I've convinced you that color information really helps us to see things quicker by using chromatic edge and surface information, and that color helps us to remember things better, and that color constancy is essential for this to discount changing illumination conditions. Thank you very much. Well, I think that one has to keep in mind that color constancy theoretically and mathematically cannot be achieved 100%. So there are conditions where there are always conditions where color constancy will fail. What these results have shown is that if you help the observer as much as possible by setting the conditions so that there is a single illuminant, there is a rich environment and everything, that observers will be able to achieve really good constancy. But you also have to keep in mind that sometimes it's the goal of the creator of the illumination to avoid constancy. For example, if you buy meat at the counter, they don't want the meat to appear the way it does when you look at it under daylight. They want it to appear very nice, and the same thing happens with fruit. And I don't know about clothing stores, but I think that's very common. And then we experience these failures of color constancy, but you have to keep in mind that these are really specifically created. There's also, color constancy is worse if you have more than one illuminant in the scene at the same time, and this also happens relatively often if you have illumination in the room, but then light coming in through windows, that can lead to problems. I'm not sure I understood how you got the 4 numbers. But what's pretty clear is that color vision evolved very differently in different species, like in reptiles and birds, or even in fish. They have many more cone photoreceptor types. And I think in each case it can be argued that the species adapted quite well to its natural environment. Now, the question why we sort of evolved to, why mammals evolved to be dichromats, and then later on part of the primates obtained this third photoreceptor, that's of course a question that can only be answered by looking at the evolutionary process and by making arguments about something that has happened 30 million years ago. That makes it a bit more complicated. It's of course what's interesting is that a single mutation 35 million years ago would have led to an immediate benefit. It couldn't have done so if there wouldn't be any, if you would need additional stages of processing. So for these monkey animals, basically having two different type of middle or long wavelength sensitive cones basically had the advantage that they were able to see these differences, whether they could immediately see them as red or from green or so. That's sort of probably a philosophical debate. The other question about sort of inserting additional types of photoreceptors, I would assume that what Jay Knights has done, and he can probably answer that question much better than I can, or maybe I shouldn't answer that question, but it's that the monkeys also immediately had that benefit of being able to make discriminations that they couldn't make before. What these stimuli appear to the monkey, that we don't know. Well, that has of course been speculated for a long time, that we would be relatively good in color constancy for the type of changes that occur in our natural surroundings, which is basically along the daylight locus from a bluish to orange direction, as opposed to illumination changes that are sometimes used in the laboratory, like to reddish and greenish. Interestingly, in the literature, there's an occasional paper every 10 years that finds such a difference, but I think that's sort of just the regular false alarm rate, because many people I know don't see these differences. There's some more recent work out of Anja Hurlbert's lab that also shows a difference with bluish illuminations, and in the experiments that we did with the objects, if we split it up in different illumination directions, that is the different filters, then we do get slightly better constancy in this yellowish direction and this bluish direction that is more along the daylight axis. But it's really, they're small. If there are differences, then they're relatively small.